Hi, I'm Stanley Otobo, a postdoc fellow from the Wellcome Sanger Institute. So today I'm going to tell you about agarose gel electrophoresis, a technique that lets us see DNA separated by size. The agarose powder is melted in a buffer using a microwave. Then it's poured into a tray. As it cools, the gel solidifies. Notice that there's a comb in place in the tray. When the gel is set and the comb is removed, it will have created little pockets called wells. The solidified gel is then placed into the electrophoresis tank and buffer is poured on top. The buffer is a conductive solution, so it can carry an electric current. The DNA samples are mixed with loading dye. The dye makes them heavier so that they will sink into the wells and also lets us track the sample as it moves through the gel. We also need to add nucleic acid stain, which will stick to the DNA and make it visible under UV light. We use a micropipette to load the DNA samples into the wells. We also load a DNA ladder into one of the wells, which we can use to measure the DNA from our samples. DNA is negatively charged, so when we turn on the electric current, the DNA begins moving through the gel toward the positive electrode. Longer pieces of DNA move slower and stay closer to the top of the gel. During the process, you can see the DNA samples migrating just by looking at the gel, because the dye we added is visible to the naked eye. Once the electrophoresis is done, we can look at the DNA itself. We will place the gel into a special machine called a gel imager. This shines UV light over the gel. Bright glowing bands should appear. Each one is a pile of DNA fragments of the same size. The ladder has DNA fragments of known sizes, so that it creates a sort of molecular ruler which we can use to measure the DNA from our samples. So that's gel electrophoresis. A gel and some electricity gives us a way to see DNA based on size.